Great. So I'm the co-director of the Synthetic Biology Center here at MIT, and I think we have a pretty broad range of companies that are represented here. So if you're not in biotech, the way to think about synthetic biology is that it's super advanced genetic engineering, like nothing you've heard of before. So in the last 20 years, it's become relatively trivial to synthesize or write megabases of DNA, millions of base pairs of DNA. Computer-aided design has completely revolutionized how we build living cells. We can take entire computer programs that were intended for software and recode it to have it run in a bacterium. So with all of these advances, you start to see the trickling of technologies across industry, many of which are involved in sustainability. And it's likely, in fact, almost certainly in the past year, that you've touched a variety of these technologies, whether you know it or not. So if you've eaten an Impossible Burger, that is technology coming from synthetic biology. There are new ways to be able to mine metals or to create sustainable chemicals or materials. New approaches to agriculture and even fundamentally where we source food have come out of the field of synthetic biology and are slowly spreading throughout the world. And so I'm going to talk today about some of the work that we've been doing that led to our team of the Climate Grand Challenges coming together and looking at how we take these technologies and apply it to agriculture. But I just want to emphasize that a lot of the research we do in my lab and at MIT in general in the field can bring sustainability solutions to a wide range of industries. So the Climate Grand Challenge project that we set up was to take this approach, this new radical way of being able to engineer living cells, and apply it to the problem of agriculture. Agriculture is both a culprit of climate change in the sense that it produces a lot of greenhouse gases, not just carbon dioxide, but also methane and nitric oxide, which are hundreds of times worse than carbon dioxide. And it also is one of the victims of climate change, where we're expecting massive declines in the yield of some of our most important crops as a result of shifts in temperature and water, and so on. And so our team is across different departments at MIT, bringing together a variety of different approaches to the problem to both decarbonize agriculture and also create crops that are able to withstand some of the changes that we know are coming down the pipeline. So we looked at this problem from the top down, imagining where are the energy and the polluting components of agriculture, and how might we approach those problems using new tools in biotechnology. One of the surprises, if you're not in that field, is the extent to which fertilizer is an important contributor to this process. If you don't make fertilizer, plants won't grow, but it takes up 30%. Just for the nitrogen component of fertilizer, 30% of the energy use goes into the production of that fertilizer, and it makes up a major component of the greenhouse gases. So in the United States, it's about 10% of greenhouse gas comes from just fertilizer production. And globally, we use between 1% and 2% of our entire energy resources just to make nitrogen fertilizer. And so we saw this starting a number of years ago, but now really accelerating as being a good place to apply some of these technologies to try to find alternative ways that we can have food crops produce their own nitrogen. So first of all, there are a lot of plants that are able to get nitrogen from the air and convert it into a form that they can eat. These are legumes or beans that, with their roots, associate with certain types of bacteria, where those bacteria take the nitrogen in air, convert it into a form where the plant can then take it up and consume it. The problem is that we eat some beans, but it's actually not the majority of where the world's calories come from. About 75% of the world's calories come from cereals, which are corn, wheat, and rice, and these crops cannot fix their own nitrogen, and they can't form these associations with the bacteria. And so the way that you deal with that as a farmer is you just put more and more nitrogen fertilizer on the crops to try to boost the yields and get as much growth as is possible. So the challenge with this is that it's an extremely polluting industry. So much of our natural gas supply goes to the production of nitrogen. Entire wars have been fought, historically the guano wars, where bat excrement was being used to fertilize crops 150 years ago. And it utilizes so much energy and produces so much greenhouse gas that it's a major problem that has to be dealt with. And there are also secondary issues with this process, including runoff, which then causes blooms of algae in the ocean, and then also overuse produces nitric oxide, which is even worse than carbon dioxide in the production. So it's absolutely required for modern agriculture. We can't do without it, but it's a huge problem at the same time. This has been recognized since the 1970s. In fact, it's been a dream of genetic engineering to be able to do something about this since then. And there are a lot of different ways that you can imagine solving this problem. You could try to take corn and reprogram it to form the associations with bacteria that legumes do. Turns out that's really hard. You could make the corn try to fix its own nitrogen, and that's even harder, and I'll show you why. And then there's some innovative solutions that are maybe thinking outside the box, like DuPont, I believe, in the 1990s had a whole project trying to make soybeans taste like corn. So we've taken all of these different approaches, and I'm going to tell you about one that's been particularly successful. First of all, what's the problem? Genetic engineering has been great. We have all these new tools in synthetic biology, but they haven't been able to solve the problem of taking the genes that are responsible for fixing nitrogen in a bacterium and just move them into corn. You know, what's the big deal? Well, there are a couple of problems with this. So unlike other genetic engineering projects, these systems require a lot of genes to work together, and they require enormous energy and material requirements that then cause a drain on the host that you move them into. These are all the biochemical numbers, but if you wanted to think about it as yourself, we also need nitrogen. We don't make our own nitrogen, we get it by eating. But if we did make our own nitrogen, we would need organs about the size of our legs entirely devoted to that process. And to make 1 kilogram of nitrogen that we could consume, you'd have to use 10 kilograms of sugar, which is like 30 cans of Coca-Cola, just for the energy content to be able to do that. And so the challenge with that is that it's a very complicated system, requires the balancing between a lot of these different components, and the bacteria are very good at chucking it out if they don't need it, and they're really, really good at shutting it down if they don't need it. So if you put a little bit of synthetic fertilizer on the soil, it completely shuts down the bacterial ability to do that. In fact, soybean production in the United States, none of the bacteria are actually used, because even though they associate with the plant, the farmers put so much artificial fertilizer down that those bacteria don't have any incentive to make their own fertilizer, and so it all comes from a synthetic source, even though those plants are able to theoretically do that. And so we started a project a decade ago where we started to use some of these tools of DNA synthesis to go into these complicated systems and start to pull them apart and understand all the biochemical interactions that are happening within them, and importantly, how to turn them on even when they don't want to be on. And we were the first lab that used DNA synthesis to reconstruct an entire nitrogen fixation gene cluster. So we never touched an organism per se. We could use the information off the computer and just rebuild that pathway from scratch. And we published a lot of papers, and being able to move the different components around, understood the process, built megabases and megabases of DNA, trying to be able to understand how to transfer it to a new organism and how to deregulate it so that the bacteria do it even when they don't want to. The other problem with making ammonia in a bacteria, or a fixed nitrogen in a bacterium that doesn't want to do it, is that the bacteria want to eat it. It's a very valuable resource. And so you have to go in and also manipulate the cell so that it sort of pukes out all the nitrogen that you want the plant to be able to use and doesn't use it in its own metabolism. And so that involves metabolic engineering, where you go in and you manipulate the metabolism of the cell so that everything that comes in out of the atmosphere gets fixed, gets exported for the plant to consume. And so this is a project that requires synthesis of large pieces of DNA, manipulating how the bacteria feed and consume these products and how they deliver them ultimately to that plant. Now, a group of my students started a company as we were starting to understand how to do this about a decade ago. This is Pivot Bio. This is one of my former students, Karsten, who's the CEO there. And they created a product over years where they went out and identified a variety of bacteria that had the ability to attach themselves to corn. And they had the genes for nitrogen fixation, but they didn't turn them on at the right time. So we applied all our tools from synthetic biology to go in and break it up so that the bacteria are both able to associate with the corn, and they're sprayed as the corn is seeded in this old version of the product, and then the roots grow, they're coated with the bacteria, and then the bacteria, whether the farmer is using synthetic nitrogen or not, the bacteria are fixing nitrogen from the atmosphere and just pumping it into the corn and providing the corn with the fixed nitrogen. With this product, it was 25 kilograms an acre, actually, and in the current version, it's now at 40 kilograms an acre. So this got very quickly adopted. So in 2018, we launched our first product. It was about tens of thousands of acres. In 2020, we had 250,000 acres. Last year, we sold 5 million acres of this product. Part of that is not just sustainability issues, but also there have been fluctuations in the global nitrogen supply, in large part because of the war in Ukraine. In fact, a couple days ago, there was a news article where there was a prisoner exchange deal where the Ukrainians were going to get back POWs, and Ukraine was going to allow Russia to transport fertilizer to Europe in response. If you don't have an idea of what 5 million acres is, it happens to be about the same size as Massachusetts. So as you drive wherever you're going to drive, just imagine a thin film of our bacteria over everything that you see. Now, there are still some problems with this microbe. So it is able to provide a lot of nitrogen at a particular growth period, but early in the process and at the end of the process, you still have to add synthetic nitrogen. And so we've been developing alternative approaches to this. One of the challenges is that we go in and we break the regulation so that it's just the bacteria just producing nitrogen all of the time. The challenge is they don't really compete well in the soil once you do that. So we've been developing alternative approaches where we use some of our tools from synthetic biology so that instead of just turning it on all of the time, we turn it on only when the plant is in need of nitrogen and when the bacteria are attached to the root. And that involves building the regulatory systems within the bacteria that can identify that. We also took a step back, and instead of working with the microbes that we happen to have around, we took a systematic approach. We took just about every bacterium you could find anywhere that we had the legal access to be able to try to manipulate, and we took just about every pathway, again, making megabases after megabases of DNA, increasingly using lab robotics in order to do it for us. And we did systematic back-to-back experiments to figure out what's the best organism, what's the best pathway, and put them together and see how they work with corn. The microbe that we love the most is Azorhizobium cholidurans. It's also referred to colloquially as green manure because you can use it as an alternative to manure. It is able to fix nitrogen, not just associated with the roots, but it forms also these nodules with the stem. So the entire plant is fixing nitrogen. It's a very complicated pathway by which it does this, requiring 63,000 base pairs of DNA, many genes, some of which are unknown. And we could go in, and every time we have a new organism, without going into the details, we have to build a lot of parts, just like any other system that you want to try to engineer. You have to build the pieces of DNA that you understand how to put them together so that you can build up a system of that complexity. And there's a lot of technology that goes into that to be able to understand how to put the pathway together, how to regulate it, and how to decouple it so that we're not engineering it. Now, one of the things that we wanted to be able to do is to make it so that the bacteria are able to receive and send signals to the plant artificially, so that with our engineered crops, they're able to communicate with our engineered bacteria. And there's a lot of precedent for how this is done. You might think of the plant in your garden as being fairly dormant, but it's actually communicating with all the other plants in the area, and it's a whole network of communication in the soil itself. And so understanding that network has been challenging, but we want to build our own signaling communication so that we can program when plants communicate and with whom they're talking to. So they have a lot of microbes in the soil, each of which is performing a different task to then create a collective agricultural system where everybody's working together. You don't have to add chemicals and fertilizer and all this sorts of stuff. So we started to build this artificial communication. For example, if a plant's being attacked by a pathogen, signal to the bacteria, hey, I need an antibiotic, or I need nutrient, or whatever, and then the microbe sends that back up to the plant. The other direction, you could have the bacteria processing information in the soil and communicating back up that you need to do something as the plant because we're detecting that there's a problem here. And then you can imagine ever more increasingly complex communication networks where bacteria, sometimes I feel like people, bacteria are passing information to each other in order to figure out the best way to deal with the stress by which they're being attacked. Okay, so in order to do this, we had to create chemical communication signals where the plant, when it needs something, it sends a chemical to the bacteria that detect that and produce whatever is required in response. So to do this, we collaborated with a group in the UK in order to put an artificial pathway into barley, which is a cereal, in order to make, on demand, a small molecule that gets excreted out of the roots. The plant does not normally make this molecule. It's produced at a very low level, but it's at a high enough concentration where then bacteria that are associated with the root, if we put the sensor for that molecule in the bacteria, they can then turn on genes that they've been programmed to turn on. And so here you can see, in this case, the bacteria have been programmed to produce light, and so they light up very dimly when the plant is making the molecule, but they stay dark but if they're not. So we could create the system in order to transfer this to our superbug, this green manure, the azorhizobium, so it's able to detect this molecule very well. And then now we can put that sensor into the microbe, and then when it's with the barley that is producing the small molecule, the bacteria that are coating the roots recognize that they're close to the root and thus should turn on the pathway, in this case turning green. And if they're in the soil, they keep the pathway off so that they can continue to survive without being affected. So the last step we did is we attached this to the nitrogen fixation pathway. So now we have an engineered symbiotic relationship between the plant and the microbe, where the plant is making a small molecule that the microbe can consume. It senses it. It then produces the fertilizer in response when it's needed. And it's able to do this in a way that doesn't produce a growth penalty to the bacteria. And we're testing how well it's going to work in the field. We've also gone the other direction, just very briefly. So this is an example of a sensor that's a microbial sensor that is responding to a small molecule that's in the soil. It then sends a signal to the plant, and then the plant root lights up when it detects that signal. And this makes it so that you can take bacteria that are perhaps more easily engineered to be sensors for certain things and have the plant respond. So finally, we're looking at being able to reapply this technology to a lot of different problems in agriculture, going down that list of what are the contributors to the greenhouse gases that are produced in agriculture, how do we apply technologies like this to that problem. Everything from fertilizer to understanding the declines in yield that are being predicted by the models like you saw in the one in the previous talk. one of the things you may not realize is we're anticipating a 40% decline in corn yields over the next 20 to 30 years as predicted by those models, meaning we'll need almost double the acreage to get the same amount of corn that we do now. And it's places like Iowa and the central United States are going to be the most affected by that. How can we fully utilize the agricultural system to create solutions to that problem? And it's not just the plant that you see above the earth, but it's also the microbes within it. 1 teaspoon of microbes has 1,500 meters of DNA, 2 meters of fungi, billions of bacteria, thousands of species that are all naturally interconnected that are contributing to the agricultural system. And we're going in and re-engineering the system from the top down, understanding the interactions, understanding how to program it so that we can distribute complex phenotypes, things that we want the system to be able to do across these different platforms. So with that, thank you, and happy to answer any questions.